Thank you, Jessica. Our final speaker in this session is Dr. Jeff Trent, who is the President and Research Director of Translational Genomics Research Institute, otherwise known as TGEN.
Thank you. Sure. All right. The Mac transition issue is always the joy. Figure out monitors. Okay. All right, thank you. So as you can see, there's been a great deal of discussion in the last few presentations. Were really exceptionally important in providing a background for both the informatics, the components that are associated with the importance of not just sample collection, but how we use this in our beginning looks at genomic information in the dog. So over the course of the discussion of this, I was asked to modify my presentation and talk about enabling precision medicine in the dog for really our hope of helping these companion animals with cancer. So TGEN is a nonprofit biomedical research institute. I'm delighted to have had the opportunity to work with really some outstanding investigators. And the three I'll really call out initially, I'll mention several over the course of this presentation, include Pat LaRusso from Yale University, phase 1 clinical trial medical oncologist, Will Hendricks from the Vogelstein Lab, now an assistant professor at TGEN, who's here in the audience, and then Chad Khanna from his work particularly around the COTC as we began our early studies together. So I'll speak about precision medicine and human cancer, a bit about the canine cancer genomics landscape. I'll begin to talk about some of the emerging tools that we've put in place and at least one opportunity in comparative oncology. So in regards to human, most of you have seen a framework slide like this that begins and ends with the patient. It requires an amount of interaction and framework and cooperation that is remarkable. Clinically, to be able to get the consent from the patient and work with the various groups from radiology and biopsy and pathology, and also from the standpoint of ensuring that it's done in a framework that has dedicated technology and infrastructure, reports are generated, and ultimately decisions are made by the treating physician. But the reality, most of you know, and certainly any of you that do these type of trials, that there are just a host of other aspects from your interaction with not just clinicians and scientists and advocates, but ways of looking at sequencing platforms, chain of custody, HIPAA, et cetera, et cetera, et cetera. And really in the sort of DC mode, I guess, having spent a decade here myself at NHGRI, you know, the Reagan Trust and Verify, there requires a remarkable amount of trust around the clinical collaboration and the patients that participate, and a fair amount of verification from the standpoint of the FDA and CMS, and we'll talk more about that as well. So TGEN has been trusted to perform a series of these large-scale human clinical trials. The Melanoma Research Alliance, about 18 different sites participating in a trial that I'll discuss in more detail. One of the pediatric oncology consortiums, 20 different sites working on particularly relapsed patients with advanced pediatric cancers. Mike Prados and his group at UCSF, funded by the Ben and Catherine Ivy Foundation for studying glioblastoma, and another with the Ivy Consortium funding again with UCSF, and Mike Prados in the lead against a series of 8 sites for enrollment. And one of the things that you'll notice is that every single one of these projects was not funded by the federal government, and I would just say that it speaks to the fact that for some of these type of projects, they're incredibly important, the way the Morris Animal Foundation has been in making a difference for to go ahead and choose to work on these. They're difficult to get funded through conventional grant mechanisms. So at TGEN, what we've been doing is not so much panel-based testing, but integrating genomic profiling in a way that you've heard, for example, in the last presentation, and we'll talk about this for the dog in a moment, with deep exome sequencing long insert whole genome sequencing, we tend to use a variety of approach that still uses Illumina that we've published, and sequence match normal DNA, which we think is critical. RNA sequencing is also critical to confirm mutations, identify oncogenic fusions, and look at differential expression and isoforms, neoantigens, a number of others. So it really requires the integration of this, and John Carpton and David Craig at our center have been extraordinarily important. Dell, through the computer company, through a philanthropic gift to TGEN's foundation, has provided us with a remarkable infrastructure to not only just take Illumina data, but run it. And in 2011, we did about 1 million CPU hours. Last year, about this time last year, we passed 1 million CPU hours per month. And this is with a cluster that does about 31 trillion calculations per second, and now has a research pipeline of being able to look and integrate all of the DNA data and the RNA data that I told you in just a little bit over 1 day. But of course, what really makes a difference is the clinical data integration and how we align that with clinical information to generate reports that can go back, fit through the process that, again, CMS and others require. And then ultimately, those share the data back, and we use groups like CA Bioportal and ICGC. But of course, for human data, for germline data, one has to be much more careful on how it's shared, and for there, we work with dbGaP and others going forward. And the last slide that I'll show on the human side is that it's taken us several years to develop clinical collaborative applications for assessing and distributing data across a federated network that is in a secure, HIPAA-compliant, cloud-enabled, web-accessible framework. So in other words, you can log in the source data being held in a HIPAA-compliant environment so that the 20 or 30 different physicians from as many different organizations are going into a location where the data is stored in a framework that hopefully provides an important source of being able to protect the data as well as to view it. Now, most of the human cases, this is the TCGA data for melanoma about to come out in press. I was privileged to be part of that team that Linda Chen and Jeff Gershenwald headed. And of course, these landscape maps show the list of genes against the number of samples. And for melanoma, not unsurprisingly, the BRAF gene, which is shown here, represents about 50% of those cases with melanoma. Now, we've focused most of our effort on those that do not have a BRAF gene, so they largely have NRAS or NF1 or neither of those. And that's been the focus of our melanoma dream team with Pat LaRusso and, again, 18 different centers that have been involved for the past almost 4 years. The phases for this project, called the Genomic-Enabled Medicine and Melanoma Trial, was the clinical development in concert with the FDA, something that for comparative oncology we're going to need to do as well. Molecular platform optimization and validation, going ahead and going through a mandated pilot clinical trial that didn't involve actually patient treatment, a formal FDA review, IND approval, and then across all of the various subgroups. So once you begin to look at this, you have to deal with both the device side and the treatment side. And just the number of people on the FDA side, probably about 30 that averaged on the phone when we would have our pre-IND meetings, gives you a sense of just how these frameworks have to be put in place. So for those of you that have done this, this is old hat, but for us it was 2,443 pages of documentation, both around the investigational new drug application, as well as almost 1,000 patients on the pre-submission alternative to an IDE, which showed the amount of verification and validation you had to do of every aspect of this to be able to put it into human testing. Well, it took us about 3 years, but on the positive side, the trial is open. It's enrolling. It's open at Yale. It's open at all 3 Mayo Clinic sites. It's open at the University of Michigan, at Indiana University, and soon to be open at 6 other centers. And we're excited to be able to move forward with this, in part because, like the MATCH trial that's just getting underway at the National Cancer Institute, it included the fact, and this is a shout out in a very positive way, to the fact that 6 different pharmaceutical companies provided 8 different targeted agents. And the only reason that list is growing because the FDA required recommended phase 2 dosing. So you'll notice that these are single agents, but soon we have 2 more, MEK-AKT and another one as well, MEC-CDK, when recommended phase of 2 dosing is approved. So these are important ways to show that industry and academia are working together. Now, the other reason now, and I'll switch completely over to canine now, that this is so important in our mind for the canine studies, particularly we're doing in melanoma, is this particular subset. In the human, we call it triple wild type, as it'll come out in the paper in Cell just recently, is really the, from deep interrogation of canine melanomas, those that are particularly mucosal, the most common form, are indeed very similar to this. And this is really where we began to look at the genome landscape, because getting ground truth in this is extraordinarily important. In the human, just as you heard for the annotation of the human versus the dog, whether you use CI Bioportal or ICGC, whatever your favorite is, about 123 studies, about 30,000 tumors that you can assess. If you look at next generation sequencing data and ignore just for this presentation studies that are only expression studies, about 10 studies have been looked at in the dog and only about 50 tumors. We were fortunate to get funded in a U grant a few years ago for a group we called the Canine Hereditary Cancer Consortium. And that allowed us to get some infrastructure started, including we decided to sequence 25 dogs across individual, some with it, like this Cocker Spaniel with melanoma, but against a series of breeds that were either national champions or had a specific phenotype we were interested in. Deafness in a border collie, this English pointer that was an AIDS champion or an Ames champion, hip dysplasia in a golden retriever, et cetera. But to be able to really put together information to be able to have like a DB SNP or Cosmic, something that you could get to if you were trying to decide that these somatic changes were truly not breed specific, because unfortunately we can't do all of our work in a single breed, but how we might be able to look through that. And these are some of the groups that we've worked with in sharing whole genome data to create with them a detailed database. And I would again call out Elaine and the postdoc we've worked with in Elaine's lab, Brian David, as being extraordinarily useful in cross-comparing data that's utilized for a lot of our studies. Now, also, we were able to develop resources like tissue microarrays, and Barb Davis, at that point, she was at Tufts, worked with us, especially around melanoma, in developing a series of these, and those become extraordinarily useful. And the Simpson and Hewitt manuscript that came out on sporadic naturally occurring melanoma in the dog that showed how overlapping the features were of canine and human mucosal melanoma gave us the opportunity to really think about this as the model we would look after. And also, when you begin to look at the fact that prior targeted analysis had compared for NRAS, the fact that it was relatively rarely mutated, same with RAF, same with KIT, and with the pathway work that's commented on by IHC in the Simpson paper and Dan Gustafson's work that included a number of observations of expression, Westerns, drug profiling, and others. It really is not a surprise that in our study, and this shows some of the whole genome sequencing on the tumors, we always do matched sets, as I talked about, normal tumor. The 2/3 of canine mucosal melanomas are quadruple wild type, no RAF, no RAS, no KIT, no NF1. So they don't have the conventional drivers that you see certainly in the cutaneous malignant melanomas. But also, just like mucosal melanomas in the human, also true with acral in the melanomas in the human, copy number variations are far exceed single nucleotide variations, and there's no UV signature. Of course, one might expect that, but nonetheless, a major difference between those that we look at. Now, this is the work of Will Hendricks, who's looked at the genomic landscape against a set of about 30 of the tumors that we've looked at with the whole genome sequence model, in which really ultimately pathways that appear to be the most notable include association with MDM2, where we saw a significant amount of amplification in the MAPK, PI3K pathways. So again, simply because there's not the conventional mutations in BRAF doesn't mean that some of these mitogenic signaling and cell cycle and apoptotic pathways are not involved. Very clearly they are, and we're just submitting this data now. It does provide therapeutic implications for the genomic landscapes, obviously coming to mind MEK and mTOR inhibition, certainly for those cases that are amplified MDM2 inhibition. The other, of course, is for those cases that are non-amplified for MDM2 and are p53 mutant, then WEE1 might be another one. And one can, again, immediately begin to think about how these clinical trials could begin to be looked at going forward as something that we think is extremely important. Now, to switch back over to the PMED applications as well, we were fortunate to begin to think about how we could work with the dogs. And one of the reasons, of course, the privacy concerns that are associated with canine patients can be lessened somewhat. And in fact, for those of us that work on this, and this is a slide that Mark Neff gave me, I mean, this really is a life comprehensively recorded. And when we do ask for information about our patients, and they are obviously the patients, then we get an amazing amount of information, including what they wore at Halloween, et cetera. But we do think that actually in some regards that this life that's comprehensively recorded and somewhat, I'll say somewhat, less comprehensively regulated should provide a more rapid way to integrate genomic and clinical information and environmental information. And we've been really particularly blessed to be able to work with the members of the Comparative Oncology Trial Consortium, Dr. Amy LeBlanc, of course, heading that now, Chad previously working with them. And the one study that we published with Melissa Paoloni now at UCSF, working on the I-SPY effort, was published in PLOS One on just trying to say, well, in the same way that, for example, the FDA actually required us for that IND to go through a non-treatment suggestion that we could actually do something within a time frame, show all the components of this. This is one of those elements where we just said, okay, can we do biopsies of multiple sites, COT centers around the country, send them all to a single component, generate this was really not a very molecularly complex study. It was just, let's just do an AFI chip and then use a series of various tools to try to come up with predictions for drugs. But nonetheless, get pathologic review, Doug Tam and his group at CSU to be able to provide information quickly. And then ultimately generate a report that could go back for a tumor board. And we were able to do that in just under 5 days against the sample set of patients for this, to just to say, okay, we can start to get some of the same frameworks together and begin to build the information on using that. Now, there was some data that we came out of, nothing particularly exciting, but for example, that there was not a clustering by breed for expression analysis. There was a clustering by tumor. That's what this suggested. And again, I don't want to make a lot out of the genomic information from this. It was really a feasibility trial for something that we thought could really make a difference going forward. Now, at TGEN, we've worked with Agilent instead of with Roche in regards to the CAM-FAM exome development. We're in the middle of that. We've also developed an in-house targeted amplicon sequencing effort with Mohammed Murtaza, one of the faculty at TGEN, and with Will Hendricks here to use with Raindance and Illumina. And we have a small gene panel of about 250 amplicons that we're using across a large set of samples, including from the CCOGC, for example. But what we're particularly excited about is the fact that we're also able to use some of the drug rule databases that are literature-derived, actively curated, lifted over to Canine to be able to come up with a set of abilities to provide information about the tumor-specific alterations that we would observe, what the drug evidence is, what the genomic alteration details are, and whether there are any available trials, and we're moving on that rather quickly. So the last thing that I'll mention in just the last couple of minutes is we are really blessed to work with a series of outstanding investigators, including Cheryl London at Ohio State, Don Berry at MD Anderson, at the National Cancer Institute, because this will be a U grant with both Paul Meltzer, Javed Khan, and then with Amy LeBlanc working on COTC, at Yale University with Pat LaRusso, at CSU with Doug Tam and Don Gustafson, and Nick Schork, who's at the Venter Institute, to come up with a framework to really attempt to see whether or not we could make a difference in a single grant cycle. So, picture how long it took us to just get the one IND approved for the one trial. is what if we could look at early, relatively treatment-naive cancers in a correlative trial. This is the sample flow for this correlative trial to look at surgical resection to no evidence of disease at COTC sites for patients with mucosal melanoma or for osteosarcoma. We came up with a factorial trial design that was developed by Don Berry, which is an adjuvant semi-adaptive trial design with correlative genomic analysis, including, for example, circulating DNA measurements and another of aspects related to predictive modeling of genomic-guided drug matching, with the goal, again, of trying to ask the question whether or not early, relatively treatment-naive cancers would respond differently than to the metastatic late-stage tumors, because most of us do believe, and certainly there's a great perspective article by Bill Haight and Arne Levine on the fact that we should really think about whether drug development in advanced human malignancies is certainly the best place that we could look for making a difference with novel agents. So with that as a background, I'll just again thank the multiple groups that we've been able to work with on precision medicine studies from, again, the Multiple Myeloma Research Foundation through groups at Pediatric Brain Tumor Foundation and UCSF, a number of others, and a number of the partnering collaborations that we have that we're really excited about. And finally, on